to make this copy. And one of the issues that we have is that DNA makes there's this thing called 5' to 3', okay? DNA has a charge to it. Because it has a charge to it, we can only replicate in one direction, all right? Well, that's a problem because what we call anti-parallel strands. You guys remember reading about anti-parallel strands? Is it so familiar at all? Okay? So what happens is you have what we call these 5' and 3' ends. It just has to do with the charge that's there. We're not going to get, you don't have to get in detail on it. But we know that in a normal DNA strand, in this case, the 5 to 3' is the top strand. The bottom strand then is the opposite, 3' on this end, 5' on the other end. Everybody good with that so far? Okay? So what's going to happen is because of that, we've got kind of this weird dance that we have to do with the two strands to get them copied. This is called semi-conservative replication because we're using each strand as a template, all right, to make the new one. So the new strand is going to have half of the old strand and the other half will be a new strand. Everybody with me on that? We're using base pair rules, so A with T and C with G as we do this. So as we go through, this is what we call the direction of replication is this way. We're unzipping the DNA at this point. This is done by an enzyme called helicase. All right, helicase's job is to unzip that and break the bonds between the A's and T's and C's and G's, okay? Once they do that, we have a second enzyme that comes in called the single-stranded binding proteins, and they bind to the two strands that have been separated to keep them apart from each other because they want to come right back together, okay? Because of the way the A's and T's and C and G work, but we got to keep those apart, all right? So we get helicase unzip, single-strand binding protein, keeping them apart, and it creates something called a replication fork and a replication bubble, okay? Now we do not have a single replication fork or bubble in replication. It would take too long with 4 billion base pairs for us to do this. So there are thousands of these bubbles and forks on the lengthened strand of the DNA. Everybody with me on that? Otherwise you can't get it done, okay? So then an enzyme, this is called the leading strand, okay? Because we can add nucleotides in a 5' to 3' direction, okay? So since we can add them from a 5 to a 3', which is what it says of that, in a continuous motion, we call this the leading strand, okay? What was that all about? I didn't even hit anything. All right, we'll go back to, my picture completely disappeared. How about that? That is wild. All right, let's get back out of here. See if I can find it.
That was such a good explanation. It's so good.
Crazy. All right, back in. So now, I have a gremlin. What is going on?
What's new in photos? I don't know. What is? You tell me. Wait for it. Wait for it. Wait for it.
It gets caught on my phone. And we have weird conversations all the time. Here's like those videos.
Why wouldn't you just freeze it?
Freeze the phone.
No, freeze the screen.
Oh, no,
All right, Mike's really hesitant to even go over here. All right, so we have the leading strand again. The problem is on the bottom strand, okay, in this case, we actually have to make the strand in the opposite direction of the replication fork. All right, so we have to go, because we always have to go 5 to 3'. So even though we're unzipping this way, this bottom strand we got to make going that way. All right, so what we have to do, what your cells do is makes it in chunks. All right, we call those chunks Okazaki fragments. All right, so what happens is in 4, this is called the lagging strand, is synthesized what we call discontinuously because it's not one long process here. Primase, that's our next enzyme. So you notice there's a lot of enzymes. We've got helicase, we got single-stranded binding proteins, we got DNA polymerase. This is actually DNA polymerase 1 if you want to get technical about it. All right, and then we've got another one called primase. All right, what primase does is finds a short little area on the strand and creates a little RNA primer. All right, remember RNA has U on it. Okay, A with U instead of A with T, and so it creates a little RNA primer on there. And then from that RNA primer, this is actually DNA polymerase 3. Do I have to know that? No. Okay, but DNA polymerase then adds bases this direction, opposite of the movement. All right? Usually this is 40 to 50 bases in size. All right? Okazaki fragment. When it reaches and touches the next Okazaki fragment, okay, the RNA primer is removed, and our next enzyme, called DNA ligase, then joins the two strands together at the phosphate and the deoxyribose sugar. And then we get an RNA primer, we go opposite direction, we remove that primer, ligase comes in, puts the two strands together. We add the primer, we move that Okazaki fragment, primer gets removed, ligase comes in and puts it together. Everybody with me on that? So it's like, even though we're going this direction, we're just going this way, like this, in chunks, all the way down in the ligand strand. Again, it's called an Okazaki fragment. Some guy named Okazaki figured it out. On the quiz, am I going to ask you about all the different enzymes? You bet I will. Okay? So you got to know what those enzymes do. There's a whole matching section on the quiz about how this thing replicates. All right? So I got a little video to show you too. See, which after this lesson, it's not like it's just going to work.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Does that help? So where the heck do we get those bases from? Cause we got bases, right? And we get these bases that we then use to replicate our DNA. Where does the A's and T's and C's and G's come from? They come from what you eat. Alright, so anytime you eat plant or animal matter, you break that down into its base nucleotides. Alright, so if I eat a carrot, I actually break down, because it doesn't matter if it's a carrot, they still use A's and T's and C's and G's, it doesn't matter. Alright, so essentially you are what you eat. Alright, when it comes down to it. You're gonna use cow DNA, you're gonna use pig DNA, you're gonna use plant DNA, you're gonna use yogurt DNA, which is bacteria, whatever you choose to eat. Alright, you're gonna break that base DNA down, and then you're gonna use those base pairs in the replication of your DNA as you go through it. Any questions on that?